Okay, when do you take your exam?
Next year, next summer.
Did you already start applying, or no? Do you have to
Not
take?
yet.
You have to take the exam first, just like
Yeah,
that. Okay.
just to see where my scores are at. One advice: make sure you record the lecture because he doesn't post the lectures.
What the fuck? Where's my phone?
Everybody in the back, hear me over
Okay,
here.
if I could record and take notes at the same time, that would be great.
All right, so welcome to General Biology,
recording now.
first day of class, very exciting. Anybody not in class yet? Today is your first class? Nice, yeah, so we'll start off with the best class. Biology is the best subject, and one of my goals for this semester is for you to leave this class agreeing that biology is the best subject. So we have a bunch of things we got to talk about today. We're going to split today about half and half. We have a bunch of like kind of housekeeping, logistics things to talk about, and then we'll actually get into a little bit of content. We're going to start every class with an outline like this that goes through the content, kind of picks out some of the important things that I want you to get right off the bat. I'm going to show you this again in a few minutes after we go through the course overview. So we're going to start with all the logistics, all that boring stuff. Then we'll come back and we'll kind of midway through, we'll kind of reset and say, okay, now we're going to start a new class, and we'll get into today's material. Some of this stuff, like CC and C, it's not going to mean anything to you yet. That's okay. We're going to get there later. I'll explain what everything means, so don't worry. So before we go any further, just a little bit about me. My name is Dr. Daniel Stern Cardinal. My last name is Stern Cardinal. It's 2 words. There's no hyphen. It's confusing. I know you're going to get it wrong. It's fine. Don't worry about it. Just Stern Cardinal. Do your best. My office is not relevant to you because I'm never there. But it's room 106A in the Douglas Bio Building, which is all the way on the other side of campus. It's literally like basically a closet. I'm never there, and if you ever need to talk to me, we'll just set up a time to talk on Zoom. So like, we're never gonna, and it's also always either too hot or too cold in that room. It's terrible, I'm never there. A good time to get me though is at the weekly review session. So we're doing 3 reviews every week. They're gonna be Tuesday and Friday morning, period 2. It's 10:20 to 11:40 a.m. upstairs around the corner in room 205. So those will be in person here. And then every Thursday afternoon from 12:10 to 1:30, we'll do a session on Zoom. That link will be on Canvas by Thursday morning. It'll just be Dr. Stern Cardinal's Zoom link. It'll be there. And we'll have the same link all semester, so you don't lose it. It's just going to be the same Zoom meeting, repeating every Thursday for the whole semester. So if you can't make any of the in-person ones, come hang out on Zoom for 80 minutes. If you ever need to get a hold of anybody from the course, have a question about anything, any concern at all, use this email address, genbio@dls.rutgers.edu. Everybody should write that down. It's also right on the front page of the Canvas page, but everybody should have it. The reason for this is that if you look around this room, you can see it's a pretty full room. There's somewhere in the neighborhood of, if everybody is here, 450-ish of you. That is approximately 1/4 of the students in Gen Bio. It's actually could be more than that. We're over 1,900 in Gen Bio this semester, split across 4 lecture sections, 2 instructors. I have 2 sections, and Dr. Beal over on Bush has 2 sections. So it's really easy if you email an individual person for that email to get lost in the shuffle and not find its way to the person who needs to see it. So a bunch of Gen Bio affiliated people have access to this inbox, and that ensures that nobody falls through the cracks, and whatever question you have, that email gets to the right person so we can address it. So always contact us using this email address, and that'll guarantee it gets where it needs to go. And then what might happen is, like, that maybe I'll respond to you with my email, right, rather than go through the Gen Bio address, but start there and make sure it gets to the right place. Okay, so we're going to talk a little bit about kind of the course design in general, and we'll talk a little more specifically about things like the grading and the workshop part of the course. You don't know what that is yet, but you will by the end of next week. You'll be familiar with it. So in terms of kind of the broader structure here, this is General Biology 115. You can think of that as Gen Bio 1. And then in the spring, you'll take 116, which is Gen Bio 2. And those courses are structured exactly the same. Obviously, they have different content, but it's really just one body of material spread out across 2 courses because it's too much to cover in a single semester. So each of these courses, you're going to get your basic biology information, like just the content that's going to form the foundation for any other course you're going to take. And that could be one of anywhere of dozens of courses, cell bio, biochem, genetics, all the way up to like ecology, evolution, plant bio, and anything in between. So we have to cover all this different content across 2 semesters so you can then go off and take whatever other course you want. But we're also, in particular the workshop part of the course, going to talk about the process of memory formation, the process of learning. We're going to see what the best practices are for learning. So you're going to develop techniques and practices that are going to enable you to successfully remember and utilize the course content. And that's going to be this semester and wherever you go after this, whether that's the 200 and 300 level biology courses, or whether you take a semester of bio and decide, you know what, that was great, not for me, I'm going to go be a history major now. Those skills are still going to transfer. So we want to make sure that you're getting the skills in addition to the actual content. And then after you take 115, you could do it in the spring with 116, you could do it in the fall, doesn't matter. You'll take the 117 course, that's Biology Research Lab. So that's the lab for Gen Bio. The lab is separate, it's a separate standalone course from the 2 lecture courses. And the lab is really interesting because this is where you get to actually do science. We're going to talk about the scientific method, we're going to look at a bunch of experiments in terms of different things that we figured out when we talk about our content. But in here, you're not actually going to get to do science, you're going to do an experiment. That's what you're going to do in the lab, so it's going to be an aquatic ecology thing where you're going to go out and sample from various bodies of water, you're going to come up with a hypothesis, you're going to collect data, you're going to evaluate your hypothesis, you're going to present your findings. So this is a really useful thing in terms of getting a feel for what that process is actually like, and also you'll learn some really useful skills in here. So some of you may know, for example, what PCR is. It's a really important process in doing molecular biology in the lab. You're going to learn how to do that. And then you'll be able to take that skill with you if you don't want to go work in a research lab at some point. Now, just as I'm talking, it occurs to me, I forgot to mention a couple things just about me just a minute ago on the About Me slide that I just skipped past. I don't know why. So let me just go back to that for a second, tell you a couple of quick things here before we move on. 2 things that I'm going to apologize for up front. One is, well, actually, it's both related to the fact that I'm from Essex County in North Jersey. So one, I'm going to mess up your names, some of you. There's just sounds, because I grew up in one of the towns where they filmed The Sopranos. If you're familiar with The Sopranos, I grew up in one of those towns. So there are sounds I can't make my mouth make, and I apologize for that. The second thing is, again, because of where I grew up, if you've ever listened to your podcasts on 1.5 speed, that's my default setting. So I have to slow that down for class. But when I get really enthusiastic about something, I tend to forget that I should not be at 1.5 speed. The problem is I get really enthusiastic about a lot of biology, so I have to catch myself a lot with that. So feel free to hey, you're going to just a little bit, just feel free to remind me about that one. And then the other thing, just in terms of my background in biology, I'm an evolutionary biologist, and also my specialty is also in genetics, which we're going to do a bunch of this semester, genetics and evolution, so I'm really excited about that. Okay, sorry for doing that out of order. Now let me go back to this. So here we are in class. This is the lecture part, and I don't like to call it a lecture. That implies I'm just talking at you for 80 minutes, which is, to be fair, a lot of what's going on here. But as you'll see as we go through, I like to have you ask questions, get feedback, make it as much of a two-way street as possible, given an environment like this, or this big. So when you come here, you come to class and actively take notes. That is the #1 most important thing. You have to show up and take notes. Okay? When you go to your workshop, which will start next week, don't go to workshop this week, it starts next week. When you go to your workshop, you have to have a hard copy of your notes. You can take notes electronically, that's fine. So laptop, tablet, whatever format works best for you. But when you go to workshop, you have to have your notes on paper. The reason for that is we used to allow like laptops in workshop, but the problem is if we say, make a flowchart or photosynthesis, what would happen if you had a laptop is people just Google it, pull the flowchart and copy it instead of like constructing it from their notes. So, okay, fair enough, no electronics in workshop, so you have to build it from your notes. So if you're doing electronic notes, just make sure you print them out, bring them to workshop so you have a hard copy. Related to that, with the note-taking, if you're taking notes on a laptop, again, totally fine. Just make sure that you are focusing on bio. You're not doing other things on your laptop. Use an example since you're sitting right in front of me. My wife and I are watching Veep right now, because I've never seen Veep, which is apparently a mortal sin. I've never seen Veep. So we're watching Veep right now. And if I was sitting here watching Veep in the front row of class, in addition to being extremely not safe for work, it wouldn't just be you wasting your time sitting here in class watching Veep. Everybody in a cone behind you would also be watching Veep instead of paying attention to whatever's going on. So electronics, all good, totally fine. Just make sure that you're focusing on bio. I got to one of my afternoon classes a few years ago early, and I was sitting in the back waiting for the class before mine to finish. And about 2 rows in front of me were 2 students watching an NBA game on a laptop. It was like, what are we doing here? But also, sure, I'll watch the NBA while I sit here and wait for the class to end, that's fine. I'm not learning anything, but okay. And the other one, and please don't be this student, but the other one a few years ago, this is my all-time favorite example of this, is somebody was playing Mario Kart, I think on their phone. And I know they were playing Mario Kart on their phone because they accidentally had the volume on and they hit a banana. And it made like a like you hit a banana and spun out sound, and everybody in the room knew exactly what just happened. So like, don't be that person if you're doing notes on electronics. Make sure you're actually just paying attention and taking notes. Okay. If you go to workshop without hard copy of notes, preparedness is part of your grade. It's a very small part of your grade, but it's free points. So just make sure you print out your notes so you get those points. Also, for future things, note-taking skills are really important. Obviously, it's going to be really important here, but for those of you that want to go to medical school, note-taking is going to be really important because let me tell you, if you think, give it a few weeks and you think the content that we're doing is dense, medical school is another level. So note-taking skills are skills that you should practice. And it's never too early to get really, really good at it. So here we are. It's also, as we'll talk about, really important for memory formation. This is going to be one of the big underlying ideas of this course. What is the process of memory formation? How can you promote that process? Taking notes is going to be part of that process. You hear what I say, you paraphrase, and you write it down. That little bit of processing is going to help you form and retain memory. So it's a really important step in the process. Just listening is not going to do it. You have to do that next step. Now, in the syllabus, which is available on Canvas, there is a component of that called the skimming list. The skimming list is really useful because it basically tells you everything you're going to be responsible for the entire semester. It's broken down by lecture topic, so there's 26 parts in the skimming list, and each one tells you 2 things. First, it tells you what the outcomes are going to be, what the learning outcomes are for each lecture. So it'll be something like sequence photosynthesis. You should be able to, tell me the process of photosynthesis, right? That's what that outcome is. The second thing that the skimming list does is it has these sections of the textbook that are relevant for that topic. So it'll be like chapter 16.1 to 16.3 and chapter 17.4, right? So we kind of mix and match, we draw different things together. So what we recommend is take a look at the skimming list before class, and then just flip through those sections of the textbook. It's going to make your life easier if you do that before class, because there's going to be like vocabulary words or basic concepts or maybe a figure. And if you just look at that stuff beforehand, it's going to make class just a little bit less intimidating, because when you hear that word or see that figure in class, it won't be the first time you're exposed to it. And if you want to go one step further, what you can do is make yourself kind of like a really bare bones outline of kind of the major ideas, so that you can kind of orient yourself within the lecture topic. So if you get lost at any point, you can kind of have that anchor, that framework that you've already prepared. And then after lecture, it's useful to go back to sections of the book where maybe you missed something or you're not quite clear on something. Use the book as a reference to then go back. Everybody at the door, come on in. There are seats. Feel free to come on in. There are seats around. They're kind of sort of in the middle of sections in some places, so you might have to like step on or over people, preferably over rather than on. You can also, there's kind of in the back, there's like kind of bench-ish things. They aren't really benches, but you can sit on them. And then of course, there are walls. So just don't block the door, make sure people can get in and out. Thank you. So I was saying with the textbook, a good way to use the textbook is as a reference after class to kind of fill in any gaps you have. Reading the textbook, not recommended. That's just reading, cover to cover every word, not going to be an efficient use of your time. Speaking of the textbook, we use the OpenStax Biology 2nd edition. This is a free textbook, which we do, one, because it's a good textbook, and two, because paying for textbooks is a scam, especially when you pay for like the 1 semester online access, giant scam, I hate it. So for this class, the textbook is free. The link is on Canvas. You can download it. It's a really nice textbook. I really like it. The only thing that sometimes I prefer our previous textbook is there are some figures from the old book that are really nice that I will often use in slides because I like the figures more. When we get into the actual content today, I'll show you a figure and how I reference the figures so you can see what it's from. If you see the figures, you want to kind of have the context for that. The old book is the Campbell Biology 10th edition book, which it's not the newest edition, so it's cheap online. And related to textbooks, one more thing, there's a website I want to warn you about. It's a very bad website called LibGen, L-I-B-G-E-N. I want you to write that down because it's very bad, and I don't want you to go to it, because on LibGen, you can download books for free. It's very bad. You shouldn't go there. It's FYI. It's LibGen, L-I-B-G-E-N. Don't accidentally go to this very bad website where you can download textbooks for free. So that's the textbook. Again, I really like it, the OpenStax. It's a good book. So I'll get into some of the course policies. There are a lot of policies for this course. The reason for that is, again, because there are almost 2,000 students in this course every fall. So if there is like some weird outlier situation that happens 1% of the time, we get 20 of those every fall. So we have to have a policy for everything. There's always gray areas, there's always exceptions, there's always stuff that comes up, so we have to have this policy to make sure that we're not just like coming up with stuff on the fly. We actually have a specific policy that's uniform and fair for everybody in the course. So in order to make sure we're all on the same page with the policy, there is an assignment on Canvas called the Policy Acknowledgement. You have to do this by noon on Friday, September 20th. There are 47 items here. The way this works is you'll have just like the statement and then like a little checkbox that says, I acknowledge this policy. You have to check that box. You have to do that for all 47 of them. You must have 47 out of 47. Don't treat this like when you click on a news article and it takes you to some newspaper's website and then they're like, we use cookies, please accept our term. Just like, yeah, just let me read the article, I don't care. Like, don't do that for this. Actually read these policies because this is what governs the course, right? So like, if you have a religious observance and you have to miss your workshop, there's policy for that. If you wake up the morning of an exam and you're vomiting, there's policy for that. But they're different, right? Having a religious observance is a different kind of absence from being violently ill on the day of whatever it is. So you have to follow the right policy to make sure you can do a makeup in each of those cases, right? So it's all going to be in there. You have to know it. So it's really important that you go through and you actually read and stick to these policies. This is also the policy document itself is available on Canvas, so you just download it, just have it. Now, this assignment is not graded, but it is required. So if you don't have it done by that Friday, any subsequent workshops starting the following week, you won't get credit for those until you do it. So the best thing to do is just do it like today, and then you don't have to worry about it. Has anyone done it already? Anyone done with their policy acknowledgement? That's awesome. Good job. I'm going to keep asking that, so by like next week, it's going to be everybody's hands will be up, and we'll be good. Okay? Now, really important, even if you don't read the policy, even if you don't do the policy acknowledgement, that policy still governs the course. So, like, those are the rules we're going to follow, those are the policies we're going to follow. Again, it's important that we do this for consistency across 4 lecture sections of almost 500 people each. So we really need to stick to that policy. It's really important. Now, in terms of evaluation for this course, we're going to have 2 exams during the semester. They are on Tuesday evenings. They're multiple choice, 70 questions each. It'll be Tuesday, October 15th, and Tuesday, November 12th. Nice thing, we won't have class on those days. So on the day of the exam, it's not like we're going to have class here and then get a little break and then we come right back for the exam. It's not going to work that way. There's no classes on those 2 Tuesdays. We just have the exam in the evening. Unfortunately, it does have to be later in the day. It's at like 7 something. And the reason for that is because we need to have everyone take the exam at the same time, so we have to do it late enough that enough rooms are available on campus to do that. So those are the regular exams, Tuesday, October 15th, and Tuesday, November 12th. And then the final exam is twice as long, it's 140 questions. That's going to be on Saturday, December 21st in the afternoon. I apologize. That's just when Gen Bio always gets assigned our exam. It's always that Saturday afternoon. Sorry, can't change it. In terms of the locations for the exam, we will send out an announcement the week before, letting everyone know where you're going to go. Obviously, if you look around this room, we can't take an exam packed elbow to elbow like this. So we have to spread us out. Some of you might be in this room, many of you will be elsewhere on campus for the exams. Just look out for that announcement. We'll give you that information when we get there. Here's the grading breakdown for the course. So the exams in total are 60% of your grade, 15% for each of the exams during the semester, and then 30% for the final. The remaining 40% of your grade is in the workshops. 10% of that is a category called engagement. It's basically just, are you on time, are you prepared, and do you participate? And there's a rubric for that in the course policy. It's very specific, so you'll see how that works. Don't worry, you're probably like, what in the world is workshop? Don't worry, we're going to get there in a little bit. We'll talk about it. And then the last 30% here are the practice questions. These are basically a clicker quiz that you're going to do every week in workshop. You don't have to buy a clicker. The clickers are there. They're going to be numbered. You're going to assign a number. Don't worry about it. And the way that's going to work is you'll do this quiz at the end of workshop every week. That total, all of them combined, will count for 30% of your grade. There are going to be 12 of them. The way that it works is the first workshop we don't count. It's just to kind of make sure you're good with the process. And then we drop the lowest of the remaining 11. So in total, 10 of those quizzes are going to count, so each quiz will be 3% of your total grade. That's how the workshop clicker practice questions are going to work. We call them practice questions because they're built from old exam questions. We take old exams and we recycle the questions for use in workshop, so they're exactly the same format, exactly the same level of detail as what you're going to see on the exams. So that's why we call them practice questions. They're representative of what you get on the big exams. So let's talk about the workshops for a little bit, because this is something that's pretty unique to general biology. Workshops are an active learning portion of the course. This is really important. One word that we don't use in Gen Bio is recitation. That implies there's like a TA at the front of the room basically doing what I'm doing, except it's a much smaller room, but it's basically working the same way. That's not what we're going to do. In workshop, it's all about you all actively doing whatever it is that we're doing that week, practicing the course outcomes. The key thing with this, when we talk about course outcomes, is that all of the course outcomes are verbs. So it's going to be things like, for today, we'll get to it in just a few minutes, sequence the process of memory formation, right? That's a verb. Compare and contrast short-term memory and long-term memory, right? We're doing things. So in the workshop, you in teams of 3 or 4 or 5 people are going to practice doing those things by making things like a table or a flow chart. You're going to present your work to everybody else. So it's an active, student-centered, student-led period where the workshop facilitator is just there to kind of keep it on the rails, but they're not actually like presenting any content to you. Okay? In addition to practicing the outcomes, we're also going to develop skills for learning, because the workshop activities are not like designed that way for no reason. We designed them the way we did because that's how learning works. These are the best practices for forming memories and then being able to retrieve those memories when you need them, like on an exam. So these activities are built the way they are specifically to make your life easier, even though I'm sure at first it'll be kind of a pain in the butt because it's going to be something new. It's going to be something you haven't done before, and it's going to be different from what you may be doing in other classes. But I promise you, there is a reason we're doing it the way we're doing it that way. So again, the workshops are going to be 40% of your grade. They do not start this week. Really important. You have 3 things on your schedule for general biology. You've got Tuesday at 3:50 p.m. You've got Thursday at 3:50 p.m. Both of those are going to be in this room. And then you're going to have a third thing at a different time in a different building. It's going to be in the bio building in either room 108 or 108A. That third thing is your workshop. Don't go to that this week. Does anybody go to that this week? You can admit it. It's okay. Yeah, I know. It's okay. A bunch of people walked over there and were like, why is there nobody here? This is weird. It's okay. It starts next week. The reason for that is twofold. One, we didn't have Monday this week, so Monday people would miss a workshop. And 2, the point of the workshop is to practice the content you get in lecture. You can't have a workshop before you've had the class. So it starts next week. You gotta bring your notes, and again, it's all about practicing the outcomes and going through the process of basically training your brain the most efficient way to learn. Now, I want to really emphasize this. It's not just like, we're going to be different and weird in Gen Bio. We designed these things the way we did because of research in neuroscience, so that like we are actually promoting the practices that promote learning. Everybody stand outside. Come on in. Don't be shy. Come on in. There's room. You're in bio. Come on in. Don't be shy. Yeah, you're good. Come on in. There are not a lot of seats left. If you're not feeling shy, there's seats right up front. Don't be shy. Come on in, everybody. You're also, obviously, there's people around the walls. It's fine. Whatever. Now, before we get into the actual content, there's just a couple more things I want to make you aware of. So first, in my notes, you're going to see terms that are in red and bold. Those are like your key terms. Those are your vocab words. You should be able to define those words. Not every time, but almost every time when I have a term in red and bold, I'm going to have a definition on screen for you. If I don't, I'm going to say it verbally. And if I say it verbally, it's probably more of an accessory thing. It's not, mission critical. But those red and bold terms, you can think of those as your vocab words. Okay? Also, paraphrasing is really important. Don't write down every word I say. Instead, paraphrase. The reason for this is that paraphrasing facilitates learning. If you do that extra processing step, you hear the words, and then you rewrite them in a slightly different, probably shorter way that preserves the meaning, that's going to help with learning. That's going to make your life easier. That's an important first step. So don't just transcribe, paraphrase. It's a really important part of note-taking. As I said before, in terms of the best way to use the textbook, it's a really useful reference tool to kind of fill in gaps, get something you missed, clarify something that's confusing, but it's not useful to just like read the textbook straight through. It's not going to be an efficient use of your time. If you are so motivated, be my guest. I'm not going to discourage anybody from like reading a lot of biology, but it's not the most efficient way to learn this material, so not recommended. Okay? And then last thing on this slide, there is a document on Canvas. If you go into the modules, modules, and then there's a bunch of different modules that have like documents attached that you can download. In one of them, there is a document called Bioetymologies that is really super useful. It's just a list of all like the Greek and Latin prefixes and suffixes and roots that form a lot of the basis for the vocabulary in biology. And if you know those, it makes your life a million times easier. So I recommend downloading that, just like having it as a reference. I'll give you an example of this. So there's a type of cell in the body. If you know what it is, don't shout it out. There's a type of cell in the body called an erythrocyte. It's the fancy name for the cell. Now, cyte, C-Y-T-E, always means cell. Anytime you see blah, 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 cyte, that's always some specific kind of cell. Does anyone know what erythro means? When it's a prefix like that. Anyone know what that means? It's a color. What do you think? It's red, yeah, erythrocyte. So the word erythrocyte means red cells. What cells do you think erythrocytes are?
Red
blood cells. Yeah. Erythrocytes is a fancy word for red blood cells. And if you break down the, I think it's Latin, it just means red cell. That's easy. So a lot of our vocabulary is going to be like that. So the bioetymologies document makes your life a lot easier. So that's highly recommended. All right, last thing before we get into the real stuff. If you're having trouble in GB, talk to us. We want everybody to succeed. There's, between my two sections, I think I have like 970-ish people between my two sections. I want to give out 970-ish A's. That is what I want to do. We're never going to like curve down or anything. I really want everybody to succeed and have, in January, the first day class in January, all of you right back here, the exact same seats. Thrilled that you got an A in Gen Bio 1 and excited for Gen Bio 2. So come to my reviews. If you can't make my reviews, go to Dr. Beal's reviews over on Bush. Hers are at different times. The content is exactly the same. Dr. Beal is wonderful. What happens is people go to her reviews and then decide they like her more than they like me, and then they start going to her lectures. That happens every semester. So use Dr. Beal's reviews if you can't come to mine. Use both, all good. Use your TAs from workshop. They're each going to have a practice session each week where they can help you work through practicing the learning outcomes. They're not going to do it for you, but they're going to help you work through it. And you don't just have to go to the session that's put on by your workshop facilitator. You can go to any of them. There's like 25 TAs that do workshop, and you can go to any one of them. So there's 25 options every week. Email us. If you have any problems, email the course. We really want everybody to succeed. One thing to note is that there are going to be like bio tutors and stuff. None of them talk to us. Nobody outside of the Gen Bio program asks us what we do in terms of what the course content is, how the exams are structured, any of that stuff. So I don't recommend any of that stuff. There's a lot of resources available to you within the Gen Bio course, so please use the resources. We are happy to help. How's everybody doing? Everybody okay? Anybody have any questions about all of that stuff I just threw at you? I know it was a lot.
I still don't know where any printer is for those electronic books. Do you have any idea where it is?
The library right over here on the other side of the ravine. You can print there. Yeah, they have a whole big bank of printers in one of the corners.
Is it free?
It's free up to a certain page limit, but it's a high enough limit that it's not going to matter.
So every student gets like $20.
What is it?
Every student gets like $20 worth of.
Yeah, and it's basically they did a study. So I was in grad school when these rules changed, and it used to be you could print a lot more, and then they made the limit lower because what they found was that the vast majority of students were printing fewer than 100 pages a semester, and then there was 0.01% of students were printing 5,000 pages. They were you can't do that anymore. So basically, as long as you're not printing multiple copies of War and Peace, you should be fine. But it's if you just go over one of the bridges here and then make a left, the library right there. Was there another handout? Yeah. Yeah. Yes, those two Tuesdays, it's 7:30 or 9 something, I forget the exact time. It's different from what it used to be. If you have a class during that time, so this is not just a bio policy, but a Rutgers policy, is that if you have a class that is regularly scheduled, that conflicts with an exam that is outside of the regular class time, the regularly scheduled class takes precedent, and you'll do a makeup exam. And again, that's a university policy, that's not us. So email us with your schedule when you get closer to the exam, we'll figure it out. Is that a hand? Yeah. Are the slides going to be available? The answer is no. And the reason for that that is take a look around you and see how many people are in this room. If we post the slides, there will be a lot more empty seats because people think they can just use the slides and not come to class. So it is highly detrimental to learning to do it that way. Now, the flip side of that is very reasonable, but I want to be able to make sure I didn't miss anything. I want to make sure that like I can fill in my gaps and whatnot. I want to check my figures that I make against the figures that you show. You are more than welcome to record, take pictures, whatever. So yeah, by all means, take it with you, but we're not going to post it before or after.
This is a similar question that they made. So for like the religious reasons for the final, if you can't make it Saturday, do you do it the following Sunday?
We'll just email us as we get closer. There's a process for it, the policy. Don't worry, we'll get you figured out. Don't worry, we got a policy for it, we'll make it work. Yes, okay, I see a bunch of hands. Yeah, all right, so we're gonna go here, and then there, and then there. Yeah. Great question. The exams, so the final is cumulative. Each of the other two exams are not. So the first exam is obviously just that material. The second exam is only going to be on the outcomes from the second unit, the second set. It builds on the first set, so you can't just forget it. But then the final is cumulative, and it's, I forget the exact breakdown, but it's going to be like 60%, 20%, 20%, right? 60% new stuff, 20, 20 for the others. Let me get back here. Yeah. Your classroom workshops. You shouldn't be able to show me your schedule after. We'll figure it out. You definitely don't have another classroom workshop. Don't worry. We'll figure it out. Oh, we'll talk about that. Don't worry. We'll get there. Don't worry. Yeah, it's in the course policy, so yes. But ask me after. It's a little more complicated than that. But ask me. Yeah. Okay, and then we're going to keep going because we're going to get behind.
I was going to ask if we were going to have any study guides or like outlines before the
We are going to have study guides and outlines galore. Don't worry. We're going to build them lecture by lecture. So by the time you get to an exam, you're going to have a stack of papers that tell you exactly what to do. We're going to talk about it. Don't worry. Okay. So let's get into the actual, we're doing on time here, pretty good. Let's get into the actual content for today. Now, like I said, everything we do in Gen Bio in terms of the structure of the course is built around what we know about how learning works. So our first actual topic is going to be memory and learning. We're going to start every class like this with an outline of the major topics for the day, and then these are going to be the outcomes, your learning outcomes for today. And the important thing with these learning outcomes is that these are going to be your exam questions. So what do these things mean? If you see CC, that means compare and contrast. So compare and contrast types of memory, short-term memory and long-term memory. And then we have seq, that's sequence, the process of memory formation and learning, and sequence, the process of long-term potentiation. So like, sticking with this as an example for a second here, we're going to say compare and contrast types of memory. A question you could get on an exam might read, compare and contrast short-term memory, long-term memory, which of the following applies to both? And then you'll get a list of things, you're going to tell us which things on that list apply to both short-term memory and long-term memory. The key thing is that the wording of the outcomes is going to be the same as the wording of the exam questions. So every outcome is telling you what a potential exam question could be. Now, there are going to be more outcomes than exam questions, so it's not going to be a perfect one-to-one, but they're telling you what the potential questions could be. Okay, so part one today, we're going to be all about memory and learning. Part two, we're actually going to take that stuff and show how the organization of the workshop mimics that learning process. We're going to see how you just overlay that process onto the workshop. And then here I'm going to show you a tool that's basically a study guide. That's the BIOS form that somebody asked about. We're going to go over it, don't worry. Okay. So here, oh, the screen's not even all the way down. That's weird. Hang on. Here we go. So, a couple things I want to point out before we even get into any content on this slide, just in terms of seeing what's going on here. So first, The slides are always going to be arranged in an outline format. So you're going to have Roman numerals, 1, 2, 3, those are going to be big headings, big sections, the first level of the outline. And then within those, you'll have subheadings, A, B, C, D, however many subheadings there are in each section. Organize your notes that way too. It's going to make your life a lot easier in terms of how the content relates hierarchically, kind of where you are with each bit of the content. Organize your notes according to this outline. My outlines and Dr. Beal's outlines are the same. We coordinate with each other. We make sure everything matches up. So if you have friends in the other sections, or for whatever reason you go to Dr. Beal's sections, it's all going to be the same. So you do whatever, it's all good, it's all going to match. Second thing I want to show you here is obviously we have a figure. This is from the old textbook. So anywhere it says Pearson figure or whatever, that's going to be, Pearson is the publisher for Campbell Biology 10th edition. So that's that old textbook. I like some of the figures. So if you want to get that reference, this is figure 49.3 in that old book, that's where you can find this. If it just says figure without the publisher, then it's the new book. Then it's the OpenStax book, and you can find it in there. Okay, so that's just, in terms of the figure references, that's how to interpret that. So today, one last thing before we get into this content, I want to be really clear today. We're just going to cover this like really bare bones. This is just a basic introduction to some of the important concepts around memory and learning. So let's get into it. We're going to start with neural development, how your brain develops. So your nervous system is circuits of neurons and supporting cells. Now, neurons, see it's red and bold. I'm not going to define it in a super technical way because we're going to spend 3 whole classes on the nervous system next semester. But for today, just know that neurons are the cells in the nervous system that communicate with each other. They're the ones that are sending and receiving the signals. Okay, so we have to generate this complex system of neurons that can talk to each other. And that talking to each other is really important for recalling memories, storing memories, forming memories. And then, of course, there are a bunch of supporting cells. They're all these other cells here, other than these yellow ones. And we're going to go through what those are next semester. We're not going to worry about them. It doesn't matter what they are for today. We really just care about the neurons today. And so during embryonic development, the overall structure of the nervous system will be laid down. So your brain, your spinal cord, your central nervous system, you have your peripheral nervous system. There's a bunch of processes that are going to govern this. We're going to talk about those processes in quite a bit of detail over the next 2 semesters. So like a lot of it has to do with gene expression and gene regulation. We're going to cover that in our second unit this semester when we talk about genetics. We're going to do a lot of that in October. Also involves signal transduction, regulation that way. We're going to do that next semester when we talk about like the endocrine system and other types of signaling within your body. Now at this point, the basic network is going to be set during embryonic development. Is that it? Are we done? We're good? Born with the brain, you're good. It's that way forever. Because the cells, you don't have any more cells, right? You'll probably know this, that like neurons, you don't grow more neurons during your life. Neurons don't regenerate if they're injured. Right? So are we done after birth? We're just set in stone? No, of course not. Throughout the rest of your life, your nervous system exhibits what's called neuronal plasticity. This is the ability to modify your nervous system after birth. And we're not talking about like growing new cells or anything. We're talking about the connections between the cells. Okay, so you're remodeling your brain. It's a highly dynamic structure where your brain is really constantly evaluating itself. What connections do I need? What can I get rid of? It's always going to be changing based on what you're doing, what's going on in your life, what are you experiencing, what are you spending time on, what are you practicing. Okay. Now, most of these changes are going to occur at structures called synapses. Who's heard that word before, synapses? Yeah, a lot of people. Good. Oh, actually, this is a useful time to ask. Who is in psych right now? Anyone taking psychology? Yeah, a lot of people. There's a lot of overlap. You're going to do what we do today. You're going to do that in psych in a lot more detail. So just be aware of that. For the psych people, you're going to get this in a lot more detail in psych. You won't be expected to apply that level of detail in Gen Bio. Just be aware of that. This is as much detail as you need to know in terms of this memory and learning stuff. Today is it, but you'll do more of it in psychology. So synapses are junctions between neurons. We're going to be a little more technical about that next semester. There's a whole lecture on synapses next semester. So we're going to be a little more technical then. But for today, it's when two neurons are talking to each other. That junction is called a synapse. Changes to synapses are activity dependent. This is the big thing I want to get across today. The changes to synapses are activity dependent. So the way I like to think about this is that memory is use it or lose it. There's two layers to this. So I want you to first look at the top half of this slide here. Okay, we have a neuron right here. This is a cell body that is receiving signals from this other neuron. The red arrows indicate the signals. And there's a bunch of words that I'm not using here because we're going to learn them in detail next semester. But if you know the word action potential, that's what that is. You don't need to know that word right now, but if you do, that's what's going on. So this neuron right here is sending a lot of signals to this neuron in the middle. So over time, due to all of those signals arriving, the brain is going to say, hey, you know what, we use that junction a lot. Let's add more synapses. Let's make more connections there so it's a stronger connection because we use it so much. This is like adding more train tracks to a really busy station. Right? So you get more trains per hour. Now down here we have another synapse right there, but you can see there are no arrows. So it costs a lot of energy to maintain that biochemical connection between those two cells. If you don't use it, over time your brain is going to be like, you know what, we can just save that money. We can just pull that back, and now we don't have to spend the money maintaining that connection. We can put that energy towards something else. So your brain is constantly doing this kind of remodeling based on the activity levels associated with different synapses. And then down here, in this bottom part of the figure, we can see another phenomenon. And it's how multiple active synapses will reinforce each other. So if you have two synapses that are active together frequently, right, two different stimuli that are always together, those are going to become associated. Right? They're going to mutually reinforce each other, and because they're firing at the same time, they're going to strengthen. Your brain's going to say, hey, we use those together all the time, so let's make sure they can operate together very efficiently. You're going to strengthen the ability of each synapse to efficiently send and receive signals. And again, that's going to be activity dependent, so it's all about use. Is everybody okay? Do you have any questions so far about the content? I forgot to mention this earlier, but if you have any questions as we go through stuff, just raise your hand. Love having questions in class. If I don't see a hand up, feel free to just like shout to get my attention. Or if you see someone's hand up and I don't see that person, just like point so I can see them. The only time where if you ask a question, we're going to say, sorry, no, is if you ask me to go back a slide. The reason for that is once I say yes to that one time, I have to say yes to it every time, and then we're never going to finish class. So if there's something that you miss on the slide and I move to the next slide, just come and see me after, and I can pull it up like on my computer and just show you so you can get the thing you missed. Because I can't go back because then we'll never finish because I have to go back for everybody. Yeah? Yes. Yes, so the question was for this bottom part where it's two different synapses, that's like two different stimuli, basically, that are occurring at the same time, that then become associated. Yeah. Yep. Good. Oh yeah, absolutely. I'll give you an example that you can use. So this is a trick for remembering things on an exam. You can do something like chew a specific flavor of gum while you're studying a specific topic, and then chew that same gum during the exam. That will help you remember the stuff, because your brain associates the information with the flavor of gum. Isn't that a neat trick? Yeah. When you're reading what? Oh, so the act of like writing becomes associated with the information that you're reading. That's a really good one. Yeah. So the comment was that you could while you're reading something, even if you're not writing words on the paper, you're just like scribbling on paper so that the act of writing becomes associated with the information that you're reading. Yeah, it's a good one. I like that. I've never heard that before, but I like that. It's good. Okay, so let's talk about memory. Now, memory is a little tricky to define. So we're going to call it an anatomical and physiological event occurring at synapses. Basically, to the extent that memory has a physical medium, that physical medium is synapses. It's connections between your neurons. Forming new synapses, physiologically, is the equivalent of forming new memories. Losing synapses is the equivalent of forgetting things. And that's basically what's going on. Now, in order to form memories, you have to have neuronal plasticity. You have to have the ability to remodel the connections in your brain. And you need to have activity associated with whatever it is you're trying to remember. So reading something repeatedly, writing it repeatedly, repeated exposure, or practice with whatever that thing is, you need some kind of activity. And the more active that activity is, the better it's going to promote, and the more efficiently it's going to promote memory formation. There are two types of memory that I want you to know some specifics about. And again, if you're in psych, you're going to do this in more detail. So what we talk about right here is what you're responsible for. Short-term memory is, as the name implies, information stored for a short amount of time, and we're talking like seconds to minutes, really, is the duration for short-term memory. And then if it's not relevant, if you're not actively working with that information that's in your short-term memory, consistently, after a few minutes, it's going to be gone. Anyone know the capacity for short-term memory? There's a few different ways people say it. Yeah, your hand, Kim, real fast. Go for it.
7 plus or minus 2.
7 plus or minus 2. Yeah. Anybody else have something a little bit different? There's a couple different ways that I've heard. There's 7 plus or minus 2, 5 to 7, approximately 7. Somewhere in there is generally accepted as the capacity for your short-term memory. So in contrast to short-term memory, we have long-term memory. Long-term memory is triggered when you basically, through repeated use, tell your brain, hey, this bit of information, this is really important. You need to hang on to this. We're gonna save this one. Right? So when you act in such a way that tells your brain you need to retain the information, that triggers the long-term memory. And then when you need it, you retrieve it into the short-term memory. So you can store the information in the long-term memory, but you can't directly use it in the long-term memory. The analogy I like for this is a computer with a spinning disk hard drive and RAM. Is everybody familiar enough with computers? Be okay with that analogy? So, the information you have on your hard drive, it's persistent. It doesn't go anywhere when you turn that computer off, but you can't directly use it from the hard drive. You have to write it into the RAM, and then you could have it in the RAM and use it, but that's temporary. When you're done with it, you put something else in the RAM, it'll overwrite what's already there. Okay, so you can think about your long-term memory is your hard drive, your short-term memory is your RAM. And yes, I know with like solid-state drives, the analogy doesn't work anymore. I know, but like just go pretend it's an old spinning disk hard drive. In terms of the duration and the capacity for long-term memory, so short-term memory, you're talking a few seconds to a few minutes, 7-ish items. For long-term memory, as far as we can tell, unlimited capacity, unlimited duration. It's a little bit, it's challenging to kind of experimentally document these things, right? But theoretically unlimited in both duration and capacity, long-term memory, in contrast to short-term memory. So remember one of our outcomes was compare and contrast short-term memory and long-term memory? So the way you would do that, the way you would practice that outcome is by making a table, short-term memory, long-term memory. And then in terms of your categories, you can have duration, capacity, fill in the different things, right? That's how you would accomplish, that's how you would practice that course outcome. So everyone see how that works? That's what we want to do here. Okay? All right. So a really important distinction is that memory and learning are not the same thing. Obviously, they're closely related. They're not the same thing. Memory is, gaining knowledge, right? Remembering things. Learning is using that knowledge in some way that benefits you, right? To decrease the likelihood of a negative outcome. Basically, memory on its own doesn't help you. Learning is the application of memory to help you do whatever it is you want to do. And as an analogy for this, I'm going to use one of my favorite movies that many of you probably have not seen because it is from before many of you were born. So old. Jurassic Park, the original Jurassic. Who's seen the original Jurassic Park? Okay, great. So you all know this one. So the thing with the original Jurassic Park is you've got the velociraptors in their pen, and the fence is electrified. And like the gamekeeper dude at the park is like, they attack the fence to try to break out. They attack the electrified fence. But they remember which spots they've attacked, and they never attack the same place twice, because they attack a spot and they get zapped, and they go, oh, don't touch that part of the fence. So the next time they try a different part of the fence. So memory is remembering which part of the fence are electrified. Learning is never attacking the same place twice. That's the distinction between memory and learning. Is everybody okay with that distinction? I know it's a subtle distinction, but it is really important because there are different practices for each one. We're going to promote both, but it's kind of different steps in the process. How's everybody doing so far? Is everybody okay? All right. Doing great. Okay, so there's a really important phenomenon here called long-term potentiation. Who has heard that word before? That is more than I expected, and that's really good, because I expected 0. So that's good. The hands are going up. Excellent. So long-term potentiation is a change to the structure of synapses that you use a lot that increases the efficiency of those synapses. It makes it easier for one neuron to send a signal to another neuron. So if you have a synapse, a connection between two cells, and you use it a lot, over time you're going to induce long-term potentiation. It just increases the strength of transmission between those two cells. The really important thing with LTP is that it is facilitated, it is both facilitated by, or I guess promoted by is a better way to say it. It's promoted by use, and it also facilitates both memory and retrieval. And so we're going to look at a flowchart in a second to explain how this works. Key thing here, activity dependent. Activity dependent. The way to promote long-term potentiation is to actively practice whatever it is you want to accomplish. Like, for example, compare and contrast, short-term memory and long-term memory. Practice making that table, that's going to promote long-term potentiation of synapses associated with those memories for that information. Okay, and that's going to be a really important phenomenon because it helps, in particular, with retrieval. In order for this to happen, you need to use the information. This is the key thing, and this is what you're going to do in workshop. You use the information. Right now, you're listening, you're taking notes, but you're not doing anything with that information. You're hearing it, you're paraphrasing it, you're putting it on paper. Great, those are all good, important parts of the learning process. But the really important parts are this, and that's going to happen after lecture. It's going to happen outside of lecture, in workshop, and on your own. So, I forgot to fix that out this morning. So let me show you kind of a flow chart here. This is the outcome for this slide. This is the sequence, the process of memory formation slide. That's what we're looking at here. Okay, so the use of information is a sorting process. Basically, how you spend your time and what you do is telling your brain how to sort the various stimuli that you have been exposed to. What are the important things that we should keep? What are the unimportant things that we should discard? Right? So if you use something, that's telling your brain it's important. You're going to keep it. If you don't use something, that's telling your brain it's unimportant, and you're going to get rid of it. Right? It's going to be in your short-term memory, and then it's gone, and it never makes it to your long-term memory. What that means, like physiologically, is that no new synapses are going to form as a result of whatever, associated with whatever stimuli or whatever information it is. Okay? So let me show you this flow chart here of the basic process of memory. Now it's going to start with the stimulus. Me talking. Right? That's a stimulus. You hearing me, that's a stimulus. Now the first place it goes is actually this thing called your sensory memory storage. Every stimuli you experience goes into your sensory memory storage, most of which you don't even consciously detect. You're exposed to just a million things every day, right? They just go right by. Okay? So the duration for your sensory memory is going to be like milliseconds to seconds. If you consciously kind of detect a stimulus, do anything with that stimulus, it's going to go into your short-term memory. The process to go from a lower level of memory to a higher level of memory is called encoding. So the fact that like you're not just sitting here half asleep, you're listening to me and processing the words I'm saying, that's enough to encode that information from your sensory memory into your short-term memory. But then again, after a few minutes, if you don't do anything else with that information, it's going to be gone. You're going to forget it. And like in this class, we've been talking for an hour. 3:50 to, yeah, we talked for an hour. And like there's stuff from the beginning of the class that you wrote down, and then you didn't do anything else with it. So it's gone. It's not in your short-term memory. It was never encoded into your long-term memory. So in order to recall that stuff, you actually have to go back to your notes, right? But with persistent use of your short-term memory, you put something into your short-term memory over and over again, over a period of time, that's going to result in encoding into your long-term memory. Great, we win. That's like the goal, right? You get the information into your long-term memory, now you have it. But remember what we said a few minutes ago. You can't use the memories in your long-term memory directly. There's one more step to actually utilizing and recalling that information, and it's retrieval back into your short-term memory. That retrieval step is really hard. And I'll give you an example of when it fails. Have you ever done this thing? I've done this. Have you ever done this thing where you're taking an exam, and there's a question, and you know the answer to that question. You know the answer. It's right there. But you can't quite get it out. You can't quite find the words to get it onto the paper. You're really frustrated. So you get out of that exam, and you immediately open the textbook, you open your notes, and you read like the first 3 words of the relevant paragraph, and all of a sudden you could like write 2 paragraphs on that question. All the information was there, you just couldn't get it back out. That was a retrieval problem. The information was in your long-term memory, but you were unable to trigger the series of synapses associated with it to retrieve it into your short-term memory when you needed it. Long-term potentiation makes that easier. That's the key thing that long-term potentiation accomplishes. It makes it easy to do that retrieval step. So how do we get the brain to do that? Well, you do this feedback loop here, encoding and retrieval. This is what you're doing when you're actively practicing with the course content. When you're making a flowchart, you're making a table, you check it against your notes, you talk to your classmates about it, you erase it, you try again. Right? That practice, that repeated active use of the information, that's going to do this loop. And that is what promotes long-term potentiation. So you get the information here, but the real learning happens outside of this room when you practice with the material. Is everybody okay with that? Good? Okay. Yeah, question?
So the reason why you like can remember it, but you can't remember it, is like you're familiar with the material that you get, but you have like a learning. That's the reason.
It's the, yeah, and going one, it's that, exactly, there's a disconnect between memory and learning, right? And the retrieval is what you really need to be able to do to accomplish learning, right? Because learning is about using it in a productive way. And to go back just a couple minutes, if you remember that, the slide with the 2 synapses with the red arrows, when the arrows get bigger when they're together, that's exactly how long-term potentiation, what that was actually showing was long-term potentiation. Just hadn't introduced the words yet. But this is the difference. Another example of this that you might be able to illustrate the point is the difference of having versus not having long-term potentiation is being able to sing a song once you hear like the opening chords of that song versus not having to hear the music in order to be able just to start singing without any other help, right? It's being able to retrieve that memory without any other stimulus helping it along. That's long-term potentiation. That's what we want to accomplish. So how do we do this? Well, it's facilitated by organization. Right? You have a coherent outline for the material, you associate different topics together, And we're going to do that all across these 2 semesters. I'm constantly going to be saying things like, and in 2 weeks we're going to do this other thing that's related to this. Or remember this thing we did last month? That was related to this in this way. So we're going to be forming those associations. Chunking is another strategy that works really well. Chunking is where instead of taking just like a list of facts, you group them in a coherent way. So that, one, there's fewer things to remember, right? You remember the chunks instead of each individual thing. And then it also helps jog your memory. If you can remember the chunk, then it helps you remember the things inside it. Classic example of chunking, phone number rhythm. Who knows phone number rhythm? What's phone number rhythm? Just do it. Ba ba ba, ba ba ba, ba ba ba. Right, phone number rhythm. That's chunking. Because it's 4 chunks instead of 10 digits. Easier to remember. Right, that helps you remember. And then the big one, I hope I'm emphasizing this enough. I'm hoping to get the point across. Activity. It's all about active practice. That's the big thing that's going to promote learning. It's all about activity. How's everybody feeling? All right, so we're going to demonstrate this. We're going to demonstrate this with a keyboard. So if you're using a laptop, close your laptop, because make it really easy to fill a keyboard if you have a laptop. So close your device, because what we're going to do, this is going to be really easy. It's a very simple demonstration, and I promise you it's easy, so it's only going to take like 20 seconds. So what we're going to do in a minute is we're going to show a blank keyboard up here. And what I want you to do, it's really important that you do it exactly the way I described. It's really important, is that you're going to start on the top left corner, and you're just going to fill in the letters on the keyboard. You're going to go across the top row. Then you're going to go to the second row. You're going to fill in the second row. You go to the third row, you're going to fill in the bottom row. Okay? So if you don't have a piece of paper out, it's good to do this on paper, or you can just try to keep track in your head. But I'm not going to give you a whole lot of time to do this, because like we look at keyboards what, hundreds of times a day, maybe over 1,000. We look at keyboards all the time, right? So it's going to be really easy for everybody, but just to demonstrate the point. So we're going to go row by row, left to right, starting at tab. So we're going to start with this key right here, fill in the letters, go across this row, then across this row, and then this one. And again, this will be really easy, so just take like 20 seconds and then we'll move on.
I can even do all of them.
I know
I
where
think the middle,
I know where F is.
the middle line is
Do it on your own, do it on your own.
first starting with the caps lock is ASDF.
I don't know, but I know I can type.
Give it like another, I'm sure you're all already like over here, so we'll just give it another like 10 seconds. We're done. And move on. All right, so how do we do? Everyone got the whole keyboard, right? Everyone show me the piece of paper where you wrote out the whole keyboard. Who got the whole keyboard? Can we get the whole keyboard? Did you get the keyboard? Well, that's embarrassing. We do these things all day long. How do we not fill the keyboard? Okay, obviously you didn't get the whole keyboard, right? If you got anything, most people are going to get like QWERTY, and then kind of stop there. If you're a gamer, you got WASD probably. Other than that, probably not much, right? Usually we get a cluster of keys up here, and then that's pretty much it, right? That's weird. We're going to do this again. This time we're going to do a little bit differently. This time, put down your pencil, grab your fingers, type the sentence, the 5 box in the Wizard's Jump quickly. Type that out.
Oh shit. I can't do that.
Who knows a keyboard now?
Nope.
Yeah, it was a lot easier, right?
It was easier, but I
It's
can't.
Go on
easier
there.
to like,
That's the difference
Physically
between
feel.
passive
yeah,
and
exactly.
active, right? That's the difference. Active learning facilitates recall. That was active, right? You don't stare at a keyboard and try to memorize a keyboard. How do you practice with a keyboard? Type. So if I ask you to type, you're going to be really good at it, right? If I ask you to fill in the letters, you never do that, right? So that was useless. This is what I want to get across for bio. Okay, so those outcomes that I gave you at the beginning, those are the exam questions. So if I say, compare and contrast short-term memory and long-term memory, make that table from memory. Practice doing it, because if you can do that from memory, you're going to be able to answer that question. Just like if I said, in order to get an A in this class, you have to be able to run an 8-minute mile. You You're not gonna like go online and like watch videos of the track events at the Olympics. You're gonna go for a run so you can succeed in the class, right? Same idea. So we want you to actively practice, and that's how you're gonna succeed with these course outcomes. So everybody kind of see how that works? Okay. Okay. So now let's talk briefly about the organization of workshop. We might get through all this, we might not, we'll see. And if we ever, just a note, if we ever don't get through everything in a day of class, that's okay. We're just gonna pick up at the next day where we left off, and like, we're never gonna miss anything. Dr. Gill and I, we write the exams. So like, we'll just coordinate, and if we decide we're behind or we've got stuff, we'll just tell you. Don't worry about it. So we're gonna get as far as we can. So, been talking about the process of learning and been kind of referencing the workshop and how activity is the key for doing these things. So it's all about active learning processes because those facilitate learning. So in the workshop, like I said, your TA is not going to be at the front of the room talking at you like I am right now. Instead, you're going to do the work. You're going to make the figures, the flowcharts, the tables. You're going to present them to each other. You're going to discuss them with each other. And your facilitator is going to be there to just kind of keep things, between the guardrails, to facilitate the activities, but they're not going to provide any of the content. By bringing your notes, you're the ones who are responsible for bringing the content into the room. Okay, so you get the content here, and then you bring it to workshop, and you use it in workshop. That's the idea. Now, the big thing you're going to do in workshop is you're going to generate organizers. Organizers are just like figures, a flowchart, a table, something like that. And the reason you're going to generate organizers is because these are tools that are going to facilitate practice, and therefore facilitate learning. The way you practice an outcome, like sequence the process of memory formation, is by making a flowchart of that process, right? You're practicing that outcome. So in workshop, that's what you're going to do. It's an active process, and therefore it promotes long-term potentiation. Okay? So that's going to be the big chunk of workshop, is the organizer creation and organizer discussion. And you're probably like, how does this work in practice? You'll see. When we do workshop next week, we're going to kind of go through it step by step with you. So don't worry if you're like not sure about what's going on with workshop yet. It's okay. You'll see next week. Now in workshop, we're also going to evaluate you. We have practice questions in there that I mentioned earlier that are the clicker quizzes that are recited exam questions. So we take the old exams and we use those for workshop questions so that you get an idea of the structure of the questions, the level of detail of the questions, how the structure of the question relates to the outcomes. You're going to see that in workshop. So that's the graded portion. And then we're not going to do this every week. It just depends on the timing, week to week. But when we have time for it, we're going to do this thing called a reality check, which is not graded. But at the end of the workshop, we'll say, okay, you made a flowchart of photosynthesis, right? You made this flowchart on a whiteboard, and then you did your practice questions. So we say, okay, blank whiteboards, take 2 minutes, make that flowchart as much as you can. And the reason we do that is a lot of times you'll make this big flowchart, and we'll say, okay, how do you feel about photosynthesis? You look at your flowchart, great, I understand photosynthesis, I got this. This all makes sense to me. And then you think you're done. You're not done, because memory formation occurs over a period of time. It doesn't occur within a single 80-minute workshop. So what we want to get across with the reality checks is, okay, you just spent half an hour doing this. Now, blank sheet of paper, can you do it from memory? No, you can't, and we wouldn't expect you to be able to. We want to make sure that you understand that, so that you understand that you need to work outside of class with the material and not just be done after you leave the workshop. That's the purpose of those reality checks. Now, when we look at our exams, actually, I'm going to skip this. This is not as important. I'll talk about this another time. I'm going to skip this one for today, so don't worry about it. Because I want to get to the BIOS form, which is really important. Now, this is a review. I've told you about this before, but the goals of workshop are improve your ability to form memories, use those memories to then learn and problem solve, And again, there's kind of two things here. There's the bio-content side of things, develop the memories and develop long-term potentiation associated with those memories, but there's also the learning skills side of things, right? Develop those best practices for learning. You can take those anywhere. Those are not specific to general biology. Those are going to apply wherever, right? Whatever you do after Gen Bio, you can take those practices with you, right? And then just, if you do stick around in bio, which I hope all of you do, and I expect most of you will, if you stick around, you're going to go to your next courses, whether it's biochemistry or ecology. There's going to be stuff that you're going to bring with you, so you don't have to relearn it when you get to those courses if you already have those long-term memories from bio. So doing this in bio will also make your life easier in whatever the next class is in your bioeducation career. So the last really big thing I want to show you today is this, the bioswarm. Now this is, it's kind of a pun. We like puns in Gen Bio. I have 2 kids. You're going to get a lot of dad jokes. I'm really sorry, but I'm actually not sorry at all. It's going to be really bad jokes. So BIOS stands for Biology, Outlines, Outcomes, and Organizers. Right there. Outlines, Outcomes, and Organizers. This is available on Canvas. You should print out 26 of these, 1 for each lecture topic. Because somebody, I forget who, early in class, because of my short-term memory, it's gone, but somebody way back at the beginning of class asked about study guides. This is your study guide. This is your semester-wide study guide. For each lecture, you're going to fill this out. And for workshops, starting with workshop number 2, you need to have 4 columns, first level of outline, second level of outline, the outcomes, the organizers column, filled in and complete when you get to workshop. It's going to be part of being prepared. The way this works is you have first level of outline, second level of outline. So like for today, this is Roman numeral 1, memory and learning. Neuronal development, neuronal plasticity, memory, right? The subheadings that I showed you in the outline, that goes in your second level of your outline. And then the outcomes column right here, these are the outcomes I give you at the beginning on that first slide, right? Where it's compare and contrast this, sequence that. So for part 1C, where it was memory, on that slide was short-term memory and long-term memory. So right here, you would put compare and contrast, short-term memory and long-term memory. That's the outcome. That is also the potential exam question, right? That's the question we could ask you. Compare and contrast this and that, which of the following applies to both, or which of the following applies to just this, right? So that outcome tells you what the exam question is going to be. And then how do we get the answer? Organizers. The organizer is the tool that you're going to make to answer that question. So if it's compare and contrast, short-term and long-term memory, you're going to make a table. So in this cell right here, you can just write table. You don't have to make the table in there, obviously it wouldn't fit, but you just write table. And those are the 4 columns you need to have ready when you go to workshop. You don't need to do this for workshop number 1. Workshop number 2 is when we're going to start saying you got to have this stuff when you get to class. So that first week of workshop, we'll come see how everything works. Now, there's a couple more things. Actually, there's stuff here, material, exam questions, the answers. There's a couple more things here. Don't start getting up yet. I still have a couple minutes. And just chill. These 2 little columns here, are you ready for the exam? This one is verified correct. Is your table correct? This one says, can you recreate that table from memory? If you can put a checkbox, a check mark in each of those columns for a given outcome, you're good. That's the goal. Because if you can recreate that table from memory, you're going to be able to answer that question on the exam. Okay, so that's our kind of way we organize everything. This kind of mirrors the outline of the course and also the learning process. Okay, so there are a couple more things in here, but I'm just going to skip this. It's not important. The summary, I just want to show you that we always end the class with a practice question. Obviously today we're right at time, so we won't go through it as a class, but as long as we have time, we'll go through the practice question as a class. But the idea here, which of the following are true for both short-term and long-term memory? 1, 2, 3, 4, and then pick your correct answer. Okay, so that's a representative type of question. This is exactly the type of thing you could expect in workshop and on an exam. Okay, so that's it for today, everybody. Thank you for all being here. Welcome back. If you have any